Muchas gracias, Fernando. Thanks so much. We're going to move next to the following presentation, which is by Maria Teresa Periñán from Mir's Group in Seville. So, Maria Teresa, please.
Hello, my name is Maria Teresa Periñán. I'm a PhD student in the Movement Disorders Laboratory at the Institute of Biomedicine of Seville. Today, I would like to share with you the results from one of our latest studies called Blood microRNAs for differentiating distinct Parkinson's disease subtypes. Parkinson's disease is the second most prevalent neurodegenerative disorder, characterized by a loss of dopaminergic neurons in the substantia nigra and intracellular protein inclusions containing alpha-synuclein. While Parkinson's disease is mainly a sporadic disease, several genes such as LRRK2 and GBA have been linked to the disease. Numerous studies have tried to identify the diagnostic biomarkers based on clinical, biochemical, or imaging aspects. Nevertheless, none of these individually predict the disease. This highlights the need to identify new disease-specific biomarkers which will increase diagnostic accuracy and complement therapeutic strategies. In this sense, microRNAs are the most studied class of non-coding RNAs which play key roles in normal cellular physiology, as well as in pathogenesis of many diseases, as in this case, Parkinson's disease. Over the last decade, several studies in different body fluids have reported the regulation of microRNAs expression in Parkinson's disease patients compared with healthy individuals, but the results have been inconclusive. As an example, here you can see that there is no overlapping microRNAs between the different studies when using plasma sample, and the same for cerebrospinal fluid. In the case of serum samples, only 3 microRNAs were reported in more than one study. For this reason, our aim was to investigate the potential of several candidate microRNAs as diagnostic biomarkers of distinct Parkinson's disease subtypes. We have used a study cohort of 64 patients with PD, of whom 13 were treatment-naive de novo PD patients without LRRK2 and GBA mutations, 30 idiopathic PD patients under dopamine replacement therapy without LRRK2 and GBA mutations, 10 LRRK2 mutated patients under dopamine replacement therapy, 11 GBA mutated patients under dopamine replacement therapy as well, and 28 controls. Our selection of microRNAs was based on previous publications. We studied 29 candidate microRNAs, and MIR165P and MIR451A were used as endogenous controls. Blood samples were collected in PAXgene blood RNA tubes to obtain total RNA, including microRNAs. This RNA was reverse transcribed and then subjected to qPCR using the custom-designed Taqman Advanced microRNA card. The relative expression levels of each microRNA was calculated using the delta-delta CT calculation. Thermo Fisher Cloud was used for the analysis of the data. A p-value under 0.05 and log of all change equal or greater than 2 were considered statistically significant. ROC curves were performed using GraphPath, and logistic regression analysis of the combined microRNAs were carried out using the RGLM function. Firstly, we evaluated changes in the microRNAs levels of the different subgroups of Parkinson's disease compared to healthy controls. Sensitivity and specificity of the most promising microRNAs were depicted by ROC curve analysis. In this regard, the combination MIR19B and MIR3613 could distinguish between GBA-PD and healthy controls, with an area under the curve of 0.75. The combination MIR1 and MIR195 showed an area under the curve of 0.92 between large 2 Parkinson's disease and healthy controls. Furthermore, MIR1 showed a fair separation between de novo PD and controls, with an area under the curve of 0.8. Contrarily, the area under the curve for MIR181C 5P was not significant between idiopathic PD and healthy controls. Afterwards, we evaluated changes in the microRNA levels between the different subgroups of patients with Parkinson's disease. The combination MIR1, MIR181C3P, MIR181C5P, and MIR214 showed an area under the curve of 0.89 between idiopathic and de novo Parkinson's disease. 95 showed a fair separation between large 2 and de novo PD with an area under the curve of 0.8. Contrarily, the area under the curve for MIR19B was not significant between GBA and de novo PD. The combination MIR1 and MIR181C5P showed an area under the curve of 0.84 between large 2 and idiopathic PD. The combination MIR19B and MIR223 showed an area under the curve of 0.9 between GBA and idiopathic PD. Finally, the area under the curve for MIR195 was not significant between GBA and large 2 PD patients. In summary, the study provides data supporting the potential of MIR1-3P to be used as diagnostic biomarker for PD at an early stage. As demonstrated by RAF oncologists in 2020, a few genes such as BDNF, GCH1 and IGF1 associated with PD pathophysiology are regulated by MIR1-3P. These target genes are involved in different biological functions like the regulation of neuron apoptotic process. Additionally, two blood microRNA signatures could be useful for differentiating idiopathic from genetic PD, thus improving the patient selection for future clinical trials. Specifically, the combination MIR1-3P and MIR181C5P could distinguish between large 2 and idiopathic PD patients. On the other hand, the combination MIR19B3P and MIR223-3P could effectively separate GBA from idiopathic PD with good ROC curve. Interestingly, Mancuso oncologists in 2019 demonstrated that these two last microRNAs, MIR223 and MIR19B, showed a significant upregulation in PD compared to Alzheimer's disease. Moreover, MIR223 showed to be upregulated considering alone those PD patients with cognitive decline compared to mild cognitive impairment subjects, suggesting a different pathogenic mechanism underlying cognitive impairment in Parkinson's disease and Alzheimer's disease. Just to sum up, further studies would be necessary to fully validate the changes in these microRNAs signatures and to establish them as reliable biomarkers for PD. Finally, I would like to thank all the members from my group and to thank you for your attention.